I'll change gears a little bit and go towards the aquatic plant side of things, where this could correlate with some of the water quality stuff that we're seeing. Like I said, nutrient poor equals low plant life. For APMs or aquatic plant management plans, it's required to sample all aquatic plants and invasive species using a point intercept survey. So a point intercept survey is basically sampling every single one of these points that you can see on the right figure. So Des Moines has 604 points, and we went to every single one of these to find the littoral zone, or where the plants grow within the lake, and I'll get to that in the next slide. Once we find out where those plants stop and when they're too deep, then we kind of just skip over some of those points. And with Des Moines Lake, there was a lot of points that were over our littoral zone. So those are the deeper areas of the lake, between like 15 and like 20, 30 feet plus in water depth. Of the total 604 points that were sampled, only 131 of those sites contained aquatic plants. As you can imagine, each lake varies with how many aquatic plants are present. This is just a summary of what all the stuff that we collected throughout the APM and what we put into our spreadsheet for analysis. So we sampled for aquatic plants at each depth, and we figured out what their maximum depth of plants are and the overall depth of colonization. So what does the plant community look like? Where are these species growing? That sort of thing. Species richness. So the more species you have and different types of species, the greater biodiversity the lake has, which is what you want. You want more species that are different than you want the same, which if you have an invasive species, for instance, that invasive species like curly-leaf pondweed or Eurasian water milfoil can disrupt that species richness and create a monoculture, which means less species diversity. Number of sites where each species was found. So at each location, we took each species, identified them to species level, and at each point we can tell how many species we found, which I'll get into that in the next slide. I'll have a photo for that. Average rake fullness at each location or point on the map, we throw a rake, and the rake is in criteria of 1 to 3. So 1 being there wasn't a whole lot of plants found, versus 3, the whole rake was covered. We can't even see the rake. There is a 0, which means we threw the rake and there was no plants there. These are getting more into statistics, which I recommend you reading the APM to kind of get more of the statistical side of these last 4. I'm not going to get into them. I do describe them in great detail in the APM. For this presentation, I'll just be going over these 2, the maximum depth of plants and overall depth colonization, and the number of sites where each species was found. So this is a figure on the right just showing where we found aquatic plants. So primarily we found them in 3 to 4 feet of water. The deepest we found them was 13 feet. So this graph shows the littoral zone of where those aquatic plants are grown. So we didn't find anything beyond 14 feet. And primarily found between 3 to 4 feet. And this is usually because once the lake drops off, the sun can't penetrate as deep down in deeper lakes like Des Moines. So it's not uncommon to find the littoral zone only being 13 feet. And you can kind of see here what types of plants that we would find within these areas. So you have your cattails, which are your emergent species, and then your wild rice. Des Moines doesn't have wild rice, but a lot of lakes around here do. And then once you get into the deeper areas, like the 3 to 4 feet, that's when you start finding your submergent plants, like your northern milfoils and your Chara species. And you can kind of see a better image here of the littoral zone that I'm talking about. So just imagine that once where that arrow meets the limnetic zone where or the open water zone, that's kind of where that 14 feet would be. And then you have all your plants, and then it drops off because there's not a light penetration within those zones. Sampling points with plants. So the species that we found primarily was variable leaf pondweed. We found that species at 72 of our sites. You can see on the right here is the distribution of where we found the species. So each dot indicates a point that was sampled on the lake. So one being the sparse, we didn't really find a whole lot of plants, is white. Two is intermediate. There was some species on the rake, but not a whole lot. That one's green. And then three, the whole rake was covered in this species, and that's red. There was some areas that were shallower that we could just look in the water without sampling with the rake. And those are called visual sightings. And that is indicated with the blue check mark. So in the APM, in one of the appendix sections, every single plant that we found will have a map associated with where we found it. And the map would just look similar to what this map looks like. It did take 2 days to sample the entire lake. So we started on July 24th and ended on July 26th of last year. On the left is just all the other plants that we found that don't include visuals. So there's 10 species here that we found with the rake or visual or with just the rake. We didn't I didn't include the visual sightings on here.